The views expressed on Science for the People are not necessarily the views of this station, its affiliates, sponsors, or advertisers.
This week on Science for the People, we're talking about the big sleep that isn't sleep at all, hibernation. We'll speak with neuroscientist Kelly Drew and biologist Frank van Breukelen about what hibernation is and what it might be able to do for us at the bedside and beyond. Welcome to Science for the People. I'm Bethany Brookshire, a science writer with Science News and Society for Science and the Public. Getting through the winter can be really hard on some people. It's dark and it's cold and everything kind of sucks. I've had several friends complain that they would hibernate the whole winter away if they could. Hibernation is one of the coolest, get it, coolest, animal adaptations out there. How does it work? And what could it do for us? To find out, I'm here with Kelly Drew, a neuroscientist with the University of Alaska Fairbanks. Kelly, thank you so much for taking the time to chat with us.
Well, thank you, Bethany. It's a pleasure to have a chance to talk about this.
Now, a lot of people probably look at an animal when it's hibernating, and they kind of assume that it's in a deep sleep, but that's actually not really the case. Can you talk about the difference between hibernation and sleep?
Sure. Yeah, so hibernation, if you look at the brain waves of a hibernating animal, it looks more like they're in a coma. They have an isoelectric cortical EEG, so their brain waves are flat. So that's very different from sleep that is characterized by very specific brainwave patterns. But we do know that animals enter hibernation through sleep, so they must be sleeping to drop into hibernation, and then from there, even if when they are in hibernation, the EEG is flat, but it's very different than a coma because it's all highly coordinated. They maintain postural control and ability to respond to touch and to sound, but it is very different than sleep.
And hibernation is specifically a type of torpor. What does it mean for an animal to go torpid?
Well, so torpor is really just a suppression of metabolism and a subsequent lowering of body temperature. And so we often use them interchangeably. Hibernation is really defined as prolonged torpor. So some animals will suppress metabolism and body temperature for less than 24 hours, and that's called daily torpor. But when they suppress metabolism, stay cold for more than a day, we call it hibernation.
And you study one of the most amazing Arctic hibernators called the Arctic ground squirrel. What is special about them as hibernators?
Well, the Arctic ground squirrel is most famous for being able to supercool. So it's been demonstrated that they can lower their body temperature as cold as -3 Celsius, so -3 below freezing, and they come out of that without any damage. So they're really remarkable at being adapted to the extreme Arctic environment where temperatures are colder than some other areas, and so they tolerate very cold temperatures, and so they get colder and they hibernate a little bit longer. All hibernating animals, if their body temperature drops below 30 C, they go through what's called the spontaneous arousal, and so they'll pop back up at fairly regular intervals, and the Arctic ground squirrel is known to have very long bouts of torpor between these interbout arousals that can last, generally about 3 weeks, sometimes even longer. A small animal like a hamster will sometimes just have, 5-day torpor bouts, 5 to 7 day torpor bouts. So those are the two things that really distinguish them, is how cold they get and the length of their torpor bouts.
And when they get cold, they also really turn down their metabolism, right?
Yeah, so all hibernating animals, at least the rodents, turn down their metabolism to about the same amount, the same extent. But they have a different threshold of how cold they can get before it alarms them to wake up. And the Arctic ground squirrel has a very low threshold as far, so they can tolerate very, very cold core body temperatures, where a hamster might wake up at 5-degree core body temperature, 5 degrees C. The ground squirrel won't wake up until about -3 C.
Now, hibernation goes in stages. Can you give us a ground squirrel's winter?
So the winter starts after a summer of getting fat, and they get into their burrows. And the first thing that happens actually before they're in their burrows is you'll see a slight gradual decline in their body temperature. And we think that this is an indication that they're turning down their metabolism. And then once they get into their burrow, they get, curled up in a nest, and they're comfortable, and they are able to sleep. And soon they will drop into this torpid state. And initially, these little periods of torpor are fairly short, maybe 3 days, and then they'll wake up and go through one of these interbout arousals. And as the winter progresses, these torpor bouts get a little bit longer until they become quite regular. And so right around November, December, January, they'll have very long, regular torpor bouts. And towards the end of February, depends on males and females a little bit, but their torpor bouts will start to shorten a little bit, and eventually they'll warm up all the way, and they'll be done for that season.
And you keep talking about these interbout arousals. Most of us probably, when we think of hibernation, I know I did, I thought of an animal just kind of dropping down into hibernation and just kind of staying there like all winter. But these animals don't do that. What is happening when they're doing these arousals?
Well, so first, if we think about hibernating bears, they don't get very cold. They don't drop below 30 C. And hibernating bears don't have interbout arousals. And it's the same is true for these fat-tailed lemurs in Madagascar. And so a study on the lemurs found that when they drop below 30 degrees C core body temperature, then they have to do these arousals. Nobody really knows exactly why. There are many reasons that may be a benefit to the animals to pop back up, and they do this. This is what kind of punctuates these bouts of torpor, where they will return to normal body temperature for about 24 hours, sometimes 48 hours, and then they go back down into torpor. And so there's many theories on the advantage of these interbout arousals. One of them is that they need to urinate. Another one is that they make glucose. Another one is that they actually do sleep during those times. So when, again, like we talked about, when they're torpid, they have flat EEGs, the brain waves, no brain waves, but when they warm back up, they show slow wave sleep. And so some people suggest that sleep deprivation actually drives these interbout arousals. Some evidence supports that, some does not. It's still fairly controversial. They replenish white blood cells. So what happens when they are torpid and cold is they have very, very low circulating white cells. So the white cells marginalize against against the vessel walls, and they get sequestered into the spleen. So there really is no immune function going on while they're torpid. So another thing that happens during interbout arousal is they reinstate their immune system. And so there's a lot of things that probably promote survival during these interbout arousals. And exactly which ones are necessary are difficult to define, because we don't really know what regulates the interbout arousal. And that's one thing that we are looking at is what is the cue, what's the endogenous natural cue that tells them, hey, we better wake up for a day before we go back to sleep or back into torpor. And so one of those things is we think it has to do with metabolism, and if they use up their glucose or make too much nitrogen and they have to reestablish a healthy equilibrium with those with the carbons and the nitrogens. And when they do that, then they warm up, or they warm up to do that, and then they go back down.
And when
answers the question.
When they do warm up, what is that like? What do they go through when they're kind of warming up for nothing?
Not a lot. So they warm up. They will sleep, but mostly they kind of tidy up their nest, fluff up their cotton, stay curled tight in a little ball. They don't eat. They may urinate a little bit. They don't typically defecate. And so it really is consistent. If you were watching them, looking at their behavior, it really does look like they warm up to sleep, because that's what they do during the interbout arousals, is they sleep. And then they go back down.
And when they do go back down, it's really impressive how down they go. You mentioned that their immune system, their immune cells kind of go and end up sticking up in the spleen, but they have almost no blood flow, right?
Yeah, it's really very low blood flow. I mean, they go from a heart rate around 200 beats per minute when they're awake and active, down to 3 to 6 beats per minute when they are in deep torpor. So that respiratory rate also goes down to about, sometimes I've held a hibernating squirrel in my hand and not seen him breathe for over a minute. So it's really hard to imagine unless you've actually seen it. There are lots of stories of even veterinarians not being able to determine if a hibernating squirrel is dead or alive.
So what is it like to hold one?
It's quite fun. It's what got me hooked on studying hibernation. As the first time somebody put a hibernating squirrel in my hands, I said, "Oh my gosh, this is amazing!" I had to figure it out. But they are. I mean, they're furry, they're cute, but they're cold, and they're very gentle. So they're wild animals, but you can hold them and cuddle them, and they don't bite. And it's interesting because when they are very cold, they're a little bit stiff. But they will also hibernate at warmer temperatures. I mean, at normal room temperature, they'll hibernate, and those are really fun because they look a little bit more like they're anesthetized. They're just kind of they're groggy. I mean, if they're in kind of deep torpor at that temperature, you pick them up and they'll kind of fall to one side and but they're just very mellow. But certainly, the colder they are, the stiffer and more tightly they're curled. And so temperature plays a big role in what they look like when they're hibernating. But for the most part, they're just very, very mellow.
So you start with this little squirrel sickle, which is what I'm going to call it. And when they come back up to body temperature, how do they do that? How do they go from -3 to 37?
Well, they work really hard at it. If you look at their metabolic rate, and they're measured by oxygen consumption, it looks like they're sprinting for several hours. And it is initiated by starting thermogenesis. So they have brown fat, brown adipose tissue that humans also have, but we don't have so much of it. Rodents have a lot, and hibernating species have a ton of it. I mean, their hearts and their necks are the rib cage is just encased with this brown adipose tissue, which is really a heater. So it's brown because it has so many mitochondria, and the mitochondria have these proteins called uncoupling proteins. And so what happens is that they generate heat. So rather than making ATP, the proton flows through these uncoupling proteins, and it's kind of like water flowing over a waterfall. It's all that energy that is just generated into heat. And so they warm up in that way. So they start, warming up around the heart, and that quickly shunts blood to the head, so they warm up their brain. So in as well as, kind of generating heat in that area, they also constrict the blood vessels down in the lower extremities. And so they do physically push the blood up to the brain. And so they warm up the front of the body first. And in fact, the rear end of the body doesn't come to full temperature and full metabolism for even 2 days later. So they really focus on the vital organs, the heart and the brain, and get those things warmed up first. And slowly and eventually they start using their legs, and which may be one reason why they don't run around and play during their interbout arousal is that their rear end it's not all that functional for several hours.
I was especially interested in your studies on adenosine receptors. Can you talk a little bit about what those are and what they do?
Yeah, adenosine is most famous for being an endogenous sleep drive. So it was first shown in cats, and people who have cats know that they nap a lot. So they found that when cats were deprived of their naps, adenosine in the brain would accumulate, and when the cat was allowed to have its nap, then the adenosine would go down again. And it was through a number of different approaches and different species. It's shown that adenosine is the endogenous sleep drive. It's not all over the brain, but in some key parts of the brain, like the basal forebrain, it accumulates, and that's what causes us to fall asleep. And then as we sleep, it dissipates, and we are able to wake up. So people are familiar with adenosine if they drink any caffeinated beverages, because the reason that caffeine wakes us up is that it inhibits adenosine receptors. And so we were interested in adenosine because we knew that animals had to sleep to hibernate. And it was actually a Japanese group that first showed it in hamsters, that if they injected a drug that stimulated adenosine receptors into the brain, they could cause a hibernation-like state, and that if they gave a drug like caffeine into the brain, it would reverse a hibernating state. And so we tried it also in the Arctic ground squirrel and got similar results, but we also found that the adenosine drug, the adenosine agonist that stimulated the adenosine receptors, that it would only work in the it would only work in the winter and not in the summer. And so we have looked then at how adenosine in the brain modulates hibernation, and what the mechanism might be that changes the response between summer and winter.
And you also study the periods of arousal. Can you tell me what happens to a ground squirrel's brain as it hibernates, and what you're looking for in terms of when it goes through arousal periods?
Well, we know that there's another class of drugs that will cause the animals to come up out of hibernation. So, these phases of the hibernation cycle, I've talked about torpor and then these interbout arousals. Well, if you look at a single torpor bout, we call it, it's got 3 phases. One of them is the entrance as they cool down. One of them is the maintenance phase, and one of them is the arousal phase. And so we are pretty sure that adenosine plays an important role to drive them down, to cause them to go down into torpor. And now we've been looking at what will cause them to come back up. And actually, the coffee question is really interesting, because the coffee-like drugs will only cause them to wake up early in their torpor bout. Late in the torpor bout, it doesn't work. And so there's something else going on that induces this interbout arousal. And while lots of people have suggested the benefit of these little interbout arousals, nobody's identified the cue in the brain or the body that causes them to do this. And so there was these drugs that were found to induce arousal that work by blocking these glutamate receptors. So glutamate is another neurotransmitter, and it has certain receptors. And if you block those receptors, it will induce arousal even late in the torpor bout. And so it happens that glutamate is a derivative of glucose. It's made from glucose. And so if you don't have a lot of glucose, you don't have a lot of glutamate. And it also is kind of a sponge for nitrogen. So when the animals are not eating, just like a person who's not eating, they break down protein for the carbons in the protein, and they generate a lot of free nitrogen. And that can build up and cause toxicity. But what glutamate does is it acts like a little sponge for nitrogen, and it turns into another molecule called glutamine. And so we thought that, we still think, and we're testing this, that the absence of glucose during these long torpor bouts and the accumulation of nitrogen might be influencing the glutamate levels. And in that way, it's the crosstalk between our nutrients and metabolism and the neurotransmitters that our brain uses to communicate with. And so we're studying the role of those pathways as potential cues that in the way that the kind of nutrient availability influences cellular communication in the brain, and how that might be the cue to wake them up. But actually what we found is it's not working in the brain, it's working outside of the brain, but it's still working through glutamate. And so now we're looking at possible sources of where glutamate could be communicating outside of the brain, and still have a lot yet to learn about that.
And you've mentioned that people cool patients after cardiac arrest, and it's being investigated for stroke. What is protective about being cold?
Well, it's what I like to think of it as a combination therapy, because there's multiple things that are protective. It reduces inflammation, so inflammation is a big part of the injury after stroke, and it reduces the generation of free radicals, reactive oxygen species that are kind of like little bullets that get let loose when tissue is injured, that causes the tissue injury to progress and get worse, a lot like inflammation. Actually, the free radicals can stimulate inflammatory responses. It also cold will attenuate many of the other cellular events that lead to cell death. One of them in the brain is the release of glutamate. Glutamate is what's called an excitatory neurotransmitter, and too much of it becomes an excitotoxin. And when the tissue is cold, the glutamate is not released, even during a stroke-like event. And so just about everything you look at in the web of events that leads to this progressive cell death after a stroke or a cardiac arrest is attenuated when the tissue is cold. And so it's really very effective at protecting metabolically vulnerable tissue after their energy supply is limited. So that's why the brain and the heart is so vulnerable, is because they're so energy demanding, and the brain most of all is energy demanding. And so when you take the blood flow away, it can't get its oxygen and nutrients. And so that causes the cells to be unstable, and pretty soon they're falling apart, and all this this cascade of events happens, and it's a progressive injury. And so if you can cool it, at the very least, it lowers their metabolic demand because things are moving more slowly. But I think even more importantly, it attenuates these specific processes that lead to the cell death.
Now, you mentioned that when ground squirrels, for example, get cool, their cellular processes in their brains, so their synapses, their dendrites, retract. What does that mean for the squirrel's behavior? Like, does it forget things? Like, how does it impact the way a squirrel functions?
Well, it's a good question, and one of the questions that come up when we talk about putting people into long-term hibernation for space travel is how much brain function are they going to lose in this process, and are they going to be able to remember how to do what they know how to do to run their spaceship, when they wake up. And all we know is in hibernating species that hibernate over the winter, they lose some memory of that they knew ahead of time before they hibernated, and they retain others. They retain the ability to recognize their buddies. And then we also know that in the Arctic ground squirrel, they actually learn better. If you're going to learn new information, they learn better after they've come up out of hibernation. So there is this resprouting of synapses that seems to be related to a peak ability to acquire new information. So those are things that have yet to be fully understood and described. And it very likely relates to how cold they get. So again, we're looking at hibernating animals that get near freezing temperatures when they hibernate, and with humans, we're talking about a much less decrease in body temperature. So it's yet to be determined. We are just starting to do this stuff in rodent experiments with laboratory rats, and looking at what just 24 hours of cooling will do to their ability to remember and acquire new information. But it is a good question.
Now, Kelly, you are a consultant for a group called SpaceWorks Enterprises. What is it, and what are you doing for them?
Well, SpaceWorks is a privately owned company its mission is to solve the problems for getting people to Mars or on space travel. And so it's largely engineers, but they also have a component that is focused on human stasis and how to protect the human astronaut during space travel. And so they have a vision of long-term human hibernation for space travel. And so I'm part of a small group that is discussing the possible application of hibernation for space travel and imagining, the problems and the solutions to these problems.
And why would hibernation be necessary or useful in space?
Well, I mean, one of the two things, one is, how much food you have to carry, so just overall payload. Another advantage of being able to hibernate is just avoiding the cabin fever effects of being in a small spacecraft for 1 to 2 years. A bigger problem, really, that NASA has identified is protection from ionizing radiation, and so it is suggested and hypothesized that cooling will protect from these things, and something else that we're just starting to investigate.
That was something I found very entertaining when I talked to a bunch of people about hibernation in space, and their response was, well, otherwise we'd just drive each other nuts.
Exactly. Can you imagine? Oh, my goodness. Right. So it'd be wonderful to be able to just hibernate the time away. It's just like a ground squirrel in winter, and they wake up and they're there.
And what would be required for a person to achieve torpor or hibernate in space?
Well, that's, some of the stuff that we're talking about that's really interesting, because there's a few physicians in the group, and some of these physicians cool people for biomedical purposes, and so they're familiar with the challenges of cooling people. And their perspective is very different than my perspective, where I see the hibernating animal that does it without any problem. And so some of the questions are, well, what do you do with the waste? First, how do you feed the person while they're cold? And secondly, what do you do with the waste that they generate? And it's so interesting because a hibernating animal, even a bear that doesn't get all that cold, doesn't eat for the whole hibernation season, and they don't defecate. But as far as we know with humans, when they're cold, they still do need some nutrient, and they still do need some way to remove waste. And so those are some of the challenges, is how to feed them. Do you put an IV line in them and continually feed them glucose? Do you just try to empty food directly into the stomach? And again, from looking at hibernation, we know that the animals don't use glucose. They don't use sugar. They don't use carbs, when they're hibernating. They use protein and fat, primarily fat, and what are called ketone bodies. And so those are some of the questions that have come up.
And we can make some animals hibernate right now, right?
Right. Well, we can induce a hibernation-like state with these drugs that stimulate the adenosine receptor. Probably hasn't been enough done to say if that is exactly hibernation. We have not looked to see if we induce it that way, if they can go about their business, their hibernation bout, without any problems. So we have induced it, and then we wake them up artificially and let them go back down on their own. So we think that we've induced it. There's a lot of fine-tuning yet to do to see if it really is completely true to hibernation.
And right now, we only cool people for, a day or two, right? We don't do it for very long periods of time.
Right, and that is actually one of the questions in medicine is how long is it feasible to cool, and does the prolonged cooling improve the benefit? And it seems as though the more severe the injury, the deeper and longer the cooling, the better. But it also seems that if people are cooled longer and, 3 days or longer, that there's more complications during rewarming. So that's the little bit that we know.
And we also
but certainly
don't
nothing.
know.
Yeah, it's nothing like cooling for 3 weeks, like in the ground squirrels, or for total of 8 months, like in a bear.
Right, and of course we don't know what we might lose, what abilities or functions or memories we might lose if we do it for a long period of time.
For a long time. Yep. So that's one of the questions.
And a lot of recent movies, and not so recent movies, really seem to enjoy the idea of hibernations in space, and they kind of run with it. What kind of movies have you seen that kind of deal with hibernation, and have any of them done it really well, or have any of them done it especially badly?
Well, I have to say I work too much to really watch a lot of movies, but I have heard good things about Passengers. That's a new movie. That it's a pretty accurate portrayal of what people envision for human hibernation. But to tell the truth, even working with the folks at SpaceWorks, I'm not sure that their vision is the same as my vision, and so there's still yet a lot to be determined. But one of the things that is consistent is in an isolated capsule, because if people are cold, their immune response is suppressed. And so they need to be isolated. And they will certainly be immobilized. So the even, how they portray it in Alien, where I think it was Sigourney Weaver was, in the pod, and the group of them were in there and then they woke up. That's pretty consistent to what we can imagine.
But what is the big limit for us right now on whether or not humans could hibernate? What is kind of stopping us from being able to do it?
Well, one of them is for very long, it's what to do, how to feed, what we need for nutrients and what we do with the wastes and how to keep the gut moving if it does need to move. What happens to the heart during rewarming. So cardiac arrhythmias during rewarming is probably the biggest risk of cooling. And they can have, problems with how the blood clots, and without a pre-existing injury, that's probably not a big problem. But I would like to see the study of human cooling to be done on healthy volunteers before we apply it to clinical scenarios. because I think that's what we really need to work out, is how to comfortably cool somebody and for how long without, side effects. But, that's we have no idea what it's going to do to cognition and, the ability to remember and to learn new things, really, until we test it.
And what about bone and muscle atrophy? I know astronauts suffer a lot of bone and muscle atrophy in space.
Yeah, so that's another good point. And that's just space travel, period, because of anti-gravity. And that's another thing about hibernation, is that they have genes that stay turned on during their inactivity that maintain bone and muscle mass. That humans, if they are on bed rest, those gene expression turns down, and so they actually have atrophy. And so how to maintain that during long-term space travel is another question that maybe is unrelated to cooling per se, but if we really understood the whole suite of things that regulate hibernation, we might be able to turn those same things on as well and maintain bone and muscle mass. But as far as we know, that's still very different than what we're looking at with regard to the adenosine drug-induced cooling.
What do you think, for the sake of human health, is going to be kind of the most important thing that we need to learn?
I think how to be cold and comfortable. So we really want people to just be comfortable while their body temperature is lowered. And I think if we can make them comfortable, we'll see huge benefits of cooling in many applications.
Well, Kelly, thank you so much for taking the time to chat with us.
Well, thank you for inviting me. I really enjoyed it.
We've linked to Kelly Drew's work and more of her information at scienceforthepeople.ca. Next, we'll be speaking with Frank van Breukelen about his hibernation work using a mammal about as far away from a ground squirrel as you can get. Get ready to meet the tenrec.
Every week on Science for the People, we take the latest in scientific progress and relate it to people, our friends, our families, our communities, and our society. And we give researchers, authors, and journalists the time to talk in depth about what matters to them. If you love science but aren't satisfied with sound bites, join us again next week for Science for the People on your local radio station or anytime online at scienceforthepeople.ca.
Welcome back to Science for the People. We've talked about Arctic ground squirrels and how hibernation might be able to help us. But there's no one way to hibernate, and it's time for us to meet the tenrec. I'm here with Frank van Breukelen, a physiologist at the University of Nevada in Las Vegas, to talk about the tenrec, what it is, and what it can tell us about the origins of hibernation. Thank you so much for being here, Frank.
Thank you.
Now let's get started with something simple yet complex. What is a tenrec?
So that's a good question. Tenrecs are a group of mammals, but there's very little known about them. There's about 35 species in Madagascar. That's going to change in the next couple of years by a few species. All but 3 of the species live in Madagascar. The other 3 live in mainland Africa. And they came to Madagascar somewhere around 50 million years ago, and then radiated out into all kinds of different sorts and forms. And so they're a really fascinating group, and there's a lot of similarities between tenrecs and what we think our ancestral mammal might have looked like, placental mammal.
And you said 35 species and soon to be a different number. Why hasn't anyone really studied these guys before?
So in Madagascar, we got lemurs. And if you're going to go to Madagascar to work on an animal, what would you work on, a lemur or a tenrec? And so I think the tenrecs were sort of ignored a little bit by the scientific community because of this incredible lemur radiation that people saw. That's not to say that there wasn't work being done. There's always been work being done, but Madagascar is a difficult place to work in, and there just isn't enough work.
And you talked a little bit about how they got ignored. Are they uninspiring? What are they like?
They're really cute. And so the thing about them is that there's all kinds of different species. So we have species that look like rats, and some that look like mice, some that look like otters, some that look like shrews, some that look just like hedgehogs. And there's species that are arboreal, living in trees, fossorial, living on the ground. There's even a species that's aquatic, and it spends its entire night in stream beds eating insects and aquatic invertebrates, and then it disappears during the day, and nobody really knows what's going on. But in terms of their study, you had this incredibly charismatic group of lemurs, and these guys weren't quite as charismatic. And so if you're going to go to Madagascar, I think people were going after the lemurs.
Well, and they breed weird, don't they?
These guys are incredible in terms of their breeding. And so what you have is, in the case of the species that I work with in the lab, tenrec caudatus, or the common tenrec, they have as many as 32 babies for sure. And there's a report from the 1920s that said 35 babies. And the average litter size is somewhat variable in Madagascar. They're found all throughout the island. And so the average litter size is somewhere between, like, say, 16 and 20, depending on where they're from.
Wow. And I heard that they actually have a really weird number of nipples to go along with that.
They do. They have, their nipples are pushed off to their side a little bit more than most other mammals. And apparently the number is 29 that people find. Of course, it varies from animal to animal.
That's a little unfair to the potentially 32 to 35 babies.
Yeah, it doesn't look like they're like pigs. Like if you look at a pig will choose a nipple and stick to that nipple throughout its development. These guys don't seem to really care. They go for whatever nipple's available.
And you have a colony of tenrecs in your lab, but you also have one in your house.
Yes.
What kinds do you have?
So in the lab, we have tenrec caudatus. And in fact, these are the only ones that are in North America, the ones that we brought in. These are just have this spectacular hibernation associated with them, which is why we're working on them. I got kind of smitten by the tenrecs, and so I had an opportunity to get some Echinops telferia, or the lesser hedgehog tenrecs. And so I have those at home. They're cute.
Do they really look like hedgehogs?
They do. It's incredible. So there's two species that look like hedgehogs, the Echinops telferia, and then another one, Setophorus, or the greater hedgehog tenrec. And they look exactly like tenrecs.
And you're interested in tenrecs specifically because you're interested in hibernation. Can you talk to me about how tenrecs hibernate? So
tenrecs hibernate, or at least hibernation has been studied in at least 4 species of tenrecs, with some, differences in terms of how well it's been studied. And what we know is in the spiny tenrecs, there is large differences in how they hibernate. So if you were to look at Echinops telferia, or the guys that I have at home, what they do is they hibernate during our winter. And they'll be torpid for most of the day, and then sometimes in the early evening they'll warm up. In the wild, they'll actually come out and bask in the sun a little bit, warm up, and then they'll forage a little, which is kind of bizarre for hibernators. They don't normally eat during the hibernation period. Setophorus, or the greater hedgehog tenrec, is probably more akin to what Kelly Drew was talking about with the ground squirrel hibernation. They seem to go torpid, remain torpid for several days, then come up for a day, then go torpid again for various bouts of hibernation during the hibernation season. In the case of our common tenrecs, it's a completely different story. These guys, during our summer, or the austral winter, because they have not shifted despite coming up into the northern hemisphere, so they're still in the southern hemisphere schedule, they'll go and start hibernating, and they can hibernate at various times of the year. We even have one animal that just recently went into hibernation already for the season, and we had births 2 days ago. So kind of crazy. But during our summer, they'll hibernate and they'll be consistently torpid. So it's not like a ground squirrel where a ground squirrel will do this interbout arousal, and then you can't really tell the difference between a ground squirrel in interbout arousal versus a summer active ground squirrel. They're both very fast. They're both very active. In the case of the tenrecs, they're always lethargic. They're just more or less lethargic during the hibernation season. So it's a very different story.
So they kind of do a seasonal laziness that is accompanied by hibernation as opposed to straight hibernation?
In the wild, what they would do is they would dig a hole, and people say they can go down as much as a meter deep. And then they bury themselves and literally take a dirt nap for 8 or 9 months, we think. There hasn't been enough field work really done to really say that well. And just recently, looking at YouTube, I discovered that they communally hibernate. No one seemed to have known that. But there was a video that somebody posted showing them collecting tenrecs for food in the middle of the hibernation season, and they pulled out 13 tenrecs out of one hibernaculum. And so in the wild, they're probably doing something a little bit different than they are in the lab, because I don't think they'd ever get disturbed in the wild. In our guys, we definitely can disturb them just by going into the room a little bit. And but again, it's not the same thing as a ground squirrel. And a ground squirrel, if I were to disturb one, they would come up out of hibernation and warm up to a very warm body temperature, about 36 degrees C. And then they would be capable of running and doing all kinds of other stuff. In the tenrecs, when we arouse them or mess around with them a little bit or keep them at pretty warm temperatures, sometimes we hibernate them at 28 degrees C. When we do that, what they do instead is that they will become less lethargic, but they're still somewhat lethargic. So they'll be ataxic. They'll be sort of wobbly. And so I've seen them drink while they're in the middle of hibernation, and they just kind of like walk up kind of stumbling and sort of lap up, but not very well. And we've seen them go back into torpor in the middle of a food dish and stuff.
In the middle of a food dish.
Exactly. They do a face plant. So.
For days, they just like go torpid just in the middle of the food dish?
Yeah, they can do that, but sometimes they move around a little bit at night, which I don't think they really do in the wild, underneath the ground, but people really don't know.
Now, these animals, you've described them as pretty wildly diverse, but you've also mentioned that they're kind of, I don't want to say primitive, but evolutionarily basal?
Yeah, so back in 2013, Maureen O'Leary's group published a paper, and it was a great paper in Science that really sort of postulated what our common mammalian ancestor would have been on the placental side. And they attributed all sorts of features to them. You know, for instance, they said that their brains would have the folds, the gyri that we see, and various other features. And if we look at our tenrecs, what we find is that they have features that are even more ancestral than this quote-unquote hypothetical placental mammalian ancestor, and features that are actually even more ancestral-like than, say, the marsupials or the pouched mammals. And so, for instance, the tenrecs that I have in the lab have a really, really small brain. They have a brain for their size that's actually smaller than that of all the monotremes or egg-laying mammals, as well as the marsupials, and their brain is smooth. And they have some very, very small regions of the brain that we always thought were incredibly important. The tenrecs also have a cloaca, which is a common urogenital opening, and so all tubes end up in the one location. And we see that in the monotremes, but not in the marsupials. And we also see that in things like birds and reptiles and amphibians and fishes.
So why does this make them important?
I think they're really important because when you talk to Kelly was talking about ground squirrels. And I view ground squirrels as probably the best hibernator there ever was, right? Core temperatures that can be as low as minus 2.9 degrees C. Pretty amazing. And they have this really, really profound hibernation. Their oxygen consumption is incredibly low. And they're really good at hibernating. I liken them to, say, the Usain Bolt of the hibernation community. And I would say that our tenrecs, particularly the ones in the lab, are more like the Homer Simpson of the hibernation community. They're not very good at it. And so if we can compare a ground squirrel versus a tenrec, we might be able to say, okay, well, these are the things that are important in order to be able to hibernate. And this is what makes you a great hibernator or not a great hibernator. There's a little bit of a misnomer in terms of hibernation. A lot of people think that hibernation is this really cute adaptation, right? An animal goes in the fall and goes to sleep and then comes back up in the spring. But it turns out that there's a tremendous amount of mortality associated with hibernation. So in ground squirrels, in some populations, you might see 20 to 50% of adult ground squirrels and as much as 70% of juvenile ground squirrels die in a given winter. And so hibernation is pretty tough on animals. Yet, I think what we're looking at when we look at a ground squirrel is probably the best hibernator, and that's come out from years and years of evolution. For whatever reason, I think our tenrecs just never really evolved to be great hibernators, and that might have something to do with the climate that they're living in and their environment that they're in. But nevertheless, they're just not very good hibernators. And comparing those two, I think we're going to actually try to tease out some of the mechanisms of hibernation.
And why exactly do tenrecs hibernate? When you think about ground squirrels, for example, if you're an Arctic ground squirrel, winter is really, really harsh. If you're living in Madagascar, life is kind of like relatively sweet, right? Why do they hibernate?
Well, first off, Madagascar is not all that sweet. It depends on where you're at in Madagascar. So Madagascar is referred to as a hyper-variable environment. So we're trying to go and do some work in Madagascar right now where it snows, not what you think of when you think about Madagascar. And the southern part of Madagascar can be very dry. But the species that we work with is actually found all throughout Madagascar. And in the north, it's either really, really hot and wet or it's hotter and wetter. Either way, there's tons of food available throughout the entire year. And yet these guys are hibernating. And so it really sort of challenges what the purpose of hibernation was. We think that animals evolved to hibernate in the tropics. And yet now we think about them in terms of that northern hemisphere bias. And we're thinking about a ground squirrel and the problems of winter with low food availability and lots of cold. But that's probably not what they evolved to hibernate in to start off with. And so it's a really great question, and nobody really knows what the answer is. But there's a good idea about, well, why be out there if you don't need to be out there? If what you can do is bury yourself underneath the ground, you probably have less risk of predation or fire or various other sort of stresses. And so you just kind of hide out until that next season you reproduce, eat a little bit in order to build up some stores, and then go back down again.
And you study these guys both in Madagascar and also in your lab. What are you looking for, and how do you do it?
So, well, we started thinking that we're going to look for something different than what we're looking for. And I was recently funded by National Science Foundation in order to do some projects. And some of those are that we're going to look at the energetics of these tenrecs. We're going to try to figure out, well, how much energy are they using, and get at some of the physiological sort of mechanisms that they use. So we're going to look at things like heart rate and tissue oxygen levels and blood pressure in the context of how much oxygen consumption they're doing. But what I'm really interested in is well, what are the costs of normal metabolism? So why are we using energy? And how does a hibernator then not use energy? And so, like much of my earlier work in ground squirrels revolved around the idea that they would turn off their protein metabolism. They would stop making proteins and stop degrading proteins. And I was interested in the mechanisms of that. We're going to do the same thing in the tenrecs, but I think I'm going to find something very different. I'm not sure that they're actually going to turn off protein metabolism so much. I think they're just so variable that they can handle the stresses of not having good protein metabolism, which is a really weird idea for me. In terms of some other work, we're going to look at kidney function. So during normal activities, some 18% of blood flow might go to the kidney. And obviously that's not going to happen when you're hibernating. So how is it that they're able to maintain kidney function? And what is it that they do? Or do they maintain kidney function? We really don't know.
Now, when you talk about the way these animals kind of regulate their metabolism, or don't, as tenrecs might not, that makes me think of the way ectotherms, so cold-blooded animals, kind of don't handle their own temperature, right? They just kind of like ride local outside temperature cycles. So are tenrecs mammalian ectotherms sometimes?
Yeah, actually, it's so weird, but it's not very far off from reality. We're seeing so many things that make us see an ectotherm-like behavior. So if I keep my tenrecs at 12 degrees C, their core body temperatures can be anywhere from 13 to 32 degrees C. And even at 13 degrees C, they can run, they can swim, which is really bizarre. That doesn't make sense in the context of mammals. We don't think of mammals as doing that. We think of mammals as requiring a warm body temperature in order to really be able to be mammalian-like. And these guys, I don't think they read the book. And so they're a little confusing for us in terms of what they're doing. And much of what they're doing is actually very sort of ectothermic-like. For instance, when we were measuring oxygen consumption, at the cooler temperatures of like 12 and 14, 16 degrees C, these guys will oftentimes go for periods of like 45 minutes with no breaths. They'll just hold their breath for 45 minutes. And then there's no compensatory ventilation. They're just not ventilating for 45 minutes. And they do stuff that looks very insect-like in terms of the way that they're breathing. Insects open up their spiracles and then allow for passive gas exchange. These guys are doing something quite similar to that, and it's got us really confused.
And a lot of scientists study hibernation and animals that go into torpor because we kind of want to learn how to make humans do it, right? We want to learn how to get to space or survive brain injury. What do you think tenrecs can teach us about human hibernation?
So I think one of the problems was that when we were looking at hibernation, we were looking at the very best hibernator and saying, I want to be like the best hibernator. But we're not the best hibernator, right? We don't hibernate, or, not really. And so if we're not going to hibernate, maybe we should look at a really lousy hibernator, at something that is just kind of just sort of barely making it in the realm of hibernation, and say, okay, well, what is it that they're doing? And maybe we can find out and sort of get the bare minimum of what you need in order to be a hibernator. So if a ground squirrel has a constellation of 100 different things that makes it a great hibernator, maybe a tenrec has 10 different things. And maybe we can look at those 10 different things and say, oh, okay, so this is what we really need to do in order to be a good hibernator, or an acceptable hibernator.
Frank, thank you so much for taking the time to chat with us.
Thank you.
We have linked to Dr. Van Breuklen's website and information about his work at scienceforthepeople.ca. You'll also find links to Twitter, Facebook, and iTunes, where you can listen to past episodes, subscribe to the show, or leave a review. You can also find our Patreon page there. And if the mood strikes, please consider supporting our intrepid podcasting crew with a monthly donation to help keep the lights on. Thanks for listening, and we'll see you next week on Science for the People.
Science for the People is produced by Rochelle Saunders and edited by Ryan Bromsgrove. Helen Quivalon is our publishing liaison. We get research help from Josh Witten and consulting support from Desiree Schell. Our frequently seen guest hosts are Marion Kilgour, Anika Hazra, and Jessica Yaros. Our theme song is called Binary Consequence, and it was written and recorded by Fractal Pattern. Science for the People is entirely listener-supported. You can find us on Patreon, where you can support us with monthly donations in any amount, or you can send us a one-time donation in any amount via the donate page of our website. Science for the People is created in partnership with the Skepchick Network, a collection of blogs, podcasts, and video content focusing on the intersection of science, popular culture, politics, and social justice. You can find out more about Skepchick at skepchick.org. The show is hosted by science news writer Bethany Brookshire and me, Rochelle Saunders.